This is the Anthrobiology podcast, hosted by me, Gabby LaPera. Every 2 weeks, I interview a biological anthropologist about what makes us, us. Hello, everyone. Welcome to another episode of the Anthrobiology podcast. Joining me via Zoom, I have Dr. Gwen Robbins Schug of UNC Greensboro. Hello, how are you?
I'm good. How are you doing?
I'm really good. Thank you so much for joining me. I know it's late your time. Well, it's 4 p.m., but if you're a morning person, that's late.
Yeah, it's been a long day of meetings and grading papers. So
yeah, and I know that you're at the end of your semester, so I'm sure it's a lot of student advising and grading and thinking about tests.
Yep.
Well, I really appreciate you coming on the show. I invited you on because I keep running into your articles in the wild. And your articles are referenced by other articles. And every time I'm like, I wonder if there's anyone interesting in the bibliography here. Your name's always in there. So I was like, you know what? It's time.
All right. It's time. We're going to get it done.
I was hoping, could you give, here's the thing is that if anyone were to visit your lab website, they would see that you do a lot of different kinds of research. I guess based on that, I kind of want you to guide the conversation in terms of what would you like to talk about? Like, what is something that you are very excited that you're doing research on right now?
Okay, well, so right now I am working on a project to analyze the human skeletal remains from a Bronze Age site in Oman. And I've been working with Kim Williams. She excavated the site between 2017 and 2019. And, she's also a bioarchaeologist, but she really enjoys the archaeology side of things at this point a little bit more. And the site has a pretty strong artifactual connection to Indus civilization. And I've been working on Indus civilization for quite a while, since about 2009. And so she invited me to come to Oman with her in 2019. No, 2018. I'm sorry. It was pre-COVID. So you know how that is.
Yeah, it's all a blur.
2018. Exactly. The before times. 2018. And so we worked together at Sultan Qaboos University, going through the human remains and pulling out things that I wanted to bring back to do additional research here at UNCG. At the time, I was at Appalachian. Well, actually, it's a complicated story. I was at UNCG, but I was on a unofficial, I was on leave of absence from App. But anyway, so I brought some things back with me, and I've been doing a lot of research on infectious disease spillover in the context of the 4.2 kABP climate change event. And we're doing isotopic analysis. We're looking at the micro artifacts in dental calculus, using scanning electron microscopy and EDX analysis. I have a tech who's working on an ancient DNA project on the dental calculus. We're doing cement emannulations. We're sort of doing a lot of different kinds of research to try and reconstruct the specific adaptations and the health consequences for the people who lived in this community in the Bronze Age, because it appears that their strategy and their health outcomes in the context of this event, not just climate change, but also sociocultural changes related to the Middle Asian interaction sphere, it appears that the strategy and the health outcomes are pretty different from most of the rest of Arabia. And so that's sort of really been a major focus for me right now.
Okay, so I just want to go through a few things because this is exactly what I meant by you do so many different types of research, which is super exciting. Yeah. So in there, I heard isotopic analysis. So for listeners, that is looking at human remain, or it can really be any remains, but in this case, human remains, and seeing if there's any isotopic differences. So like if there's any differences in the, I've done so many episodes about isotopic analysis and I should have this straight, in the differences in the numbers of protons.
Yeah. So isotopes are variants of elements. And what makes an element a specific element is the number of neutrons. And so if you have a different number of protons, or wait, did I just say that wrong? Oh gosh, now we're going to have to back up. Hold on.
No, it's totally fine. I do this literally every single time I talk about isotopic analysis. I'm like, is it neutrons or is it protons? I think it might be neutrons. Yeah. I think you're right.
They have the same number of protons, but different numbers of neutrons.
Right.
Yes, because that's what makes their mass different, because protons don't really weigh anything.
Right. And then the neutrons, it's heavier or lighter. And like based on that, you can understand different things about diet. So for example, there's carbon 12 and carbon 13. And
they have the same chemical properties,
right?
But they fractionate in the body a little bit differently because of differences in weight, or they fractionate differently in water sources or whatever, because of the different atomic weight.
If you've ever watched Bones and they're like, oh, this person has they always pick one that they always end up mispronouncing, and I can't remember. Strontium.
Yes.
They always wildly mispronounce that. I don't know why.
Yes. So I'm not an expert on isotopes, but I have a PhD student, Sangeeta Mahajan. She joined from India. She actually is an amazing person. She was an oral surgeon for 30 years. And then she decided to pursue her interests in ancient Indian history and archaeology. So she went to Deccan College, which is where I did my dissertation research alongside Veena Mushri Tripathi. We literally sat like at the same table and did our research for our dissertations. But anyway, Sangeeta went there and she got 2 master's degrees, one in ancient Indian languages and literature, and then one in archaeology. And she became an expert in isotopic analysis, looking at carbon and nitrogen isotopes to infer information about diet in a population of people from Inamgaon, which they lived in what's called the Deccan Chalcolithic, about like 1500 to 1000 BC. There was a period of environmental change during this time. And these people have been trying to look at isotopes in this population for decades and have been completely unsuccessful because of the preservation and also because of like glue that was applied to the archaeological remains when they were excavated. But Sangeeta did it. And so she was able to actually get data. And so I had written to her just to congratulate her with her publication of her master's thesis results. And then fortunately, over time, we developed a relationship and she decided to come here. And so now she is working on strontium and oxygen isotopes, as well as carbon and nitrogen, for the tomb and the bone pit at the site of Dahua in Oman, which is the site that I've been really fascinated by.
Yeah.
And so while I'm not an expert on isotopic analysis by any means, she's bringing her expertise into the lab to work on that, because a lot of the populations that existed in Arabia during the Bronze Age, one of their strategies for coping with changing environments, both climatic and like socioeconomic environments, was to increase their levels of mobility. And so that was one of our sort of hypotheses, because at the site, there are so many Indus artifacts, which is unusual for these tombs in Arabia at this time. The artifacts, the iconography, there's Indus script.
To be clear, it's because they're far apart from each other.
Well, they're not super far apart, right? They're across the Arabian Sea from one another. Mm-hmm. They're within the same Middle Asian interaction sphere. So they were in a big trade network with one another.
Okay.
And we knew that there was contact and exchange going on already. But one of our questions is, if the people at this site are participating much more in these trade interactions than other sites in the peninsula, just based on the artifactual record. Is that because folks are coming from Indus civilization and settling at the site and then mining and producing copper to take back to Indus cities? Or is it that folks are coming from all over the Middle Asian interaction sphere? Or are these local folks who've gotten involved in this trade relationship and are living at this site? And so that's one of the questions that she will be investigating.
Yeah. And so one of the ways that you can use isotopic analysis is because different groups of people, especially if you're looking at like dietary differences, different groups of people eat different things, and you can like measure various differences. So I grew up in academically in the Tidewater in Virginia. So we were looking at the differences in corn consumption. So people who were newly arrived from England had very little evidence of corn. People who'd been here for a little bit longer, middling, people who grew up here a lot more. You can do the same thing in other places. You can do the same thing with nitrogen, which gives you a better idea of kind of like proteins that people might have consumed. But this is only one of the many things that you mentioned, because you also mentioned dental analysis, right? So you're looking at both like where and then also like what all is stuck up in there.
Well, so I was trained by John Lucas. I was in his lab at the University of Oregon. And so dental anthropology is like part of my, one of my great loves. And so what we're doing really, so I have to back up a little bit, because the folks who were living at this site not only are participating in this trade relationship to kind of an unprecedented degree for people in Arabia at this time, we also are trying to figure out like whether they had shifts in their diet during this period of climate change, like you mentioned with the carbon and nitrogen. But there's definitely evidence for disease spillover related to their participation in this network.
Can I actually ask you, what is disease spillover?
What we mean by spillover is basically just that new diseases are entering the population and then thriving there. So what we're finding is that infectious pathogens accompanied people into this area with this trade. And so we're finding evidence for the similar kind of infectious pathogens that we see in Indus civilization in Oman as well, at this one site. And so we're actually doing micro CT analysis on the I forgot to mention that on the maxilla and mandibles, on the maxilla and mandibles and tibiae, and potentially on the eye orbits for individuals from the site, because they're very fragmented. And we would really like to get a good sense of what kinds of osseous changes are happening on a microstructural level because of the introduction of these pathogens. But to get back to the calculus, a lot of these folks had really, really significant periodontal infection. And so there's a lot of alveolar bone loss. The surface of the tooth roots is exposed to the oral environment. And so there are, for this region, fairly significant amounts of dental calculus, which is just mineralized plaque on the surface of the teeth. And so one of the things we've been doing is looking at it in the scanning electron microscope to try and identify artifacts, food refuse, as well as occupational debris in these people's teeth, in the calculus.
I think a lot of people don't realize, especially in the United States, people with access to dentistry don't really realize like how thick and how much calculus can build up on the teeth. So it can be like millimeters thick. And so when you talk about catching food refuse or like occupation related refuse, like there's like actual particles. It's not like someone died and they were like eating a carrot and we found the carrot. It's like actual food particles can be embedded in this material.
Right. So there's cellulose from plants that are being consumed, as well as starch granules. There are fibers of collagen from animals that are being consumed. And there's also bacterial cellulose from these bacteria that are creating the biofilm that turns into this mineralized plaque, we find evidence not only for the lattice that the bacteria created when they were occupying the dental calculus, but in some cases, we can actually see the bacterial cellulose and preserved bacteria as well, because sometimes the bacteria calcify. And so they themselves are actually still preserved in the dental calculus.
That is so cool.
We find evidence of yeast. We find occupational debris like fibers, pieces of marine organisms. We find food refuse, like I mentioned, the collagen and the cellulose and starch granules. And so we're just using SEM to sort of identify what's there, and then we'll be doing a phytolith analysis. In some cases, you can actually identify plant parts, at least to genus, in the scanning electron microscope. So you can see their little silica skeleton pieces in the scanning electron microscope, but a lot of them are easier to identify using light microscopy. So we'll actually, after we're done doing the SEM on the dental calculus, we'll also take some samples and look at them in the light microscope. But to do that, you actually have to destroy the calculus. So we're a little bit more hesitant about that.
That is something that I don't think has come up a ton on this podcast. It has some, which is that sometimes the only testing that we can do is destructive. And so you have to consider carefully whether or not you want to do that, and if what you gain will be worth it, especially keeping in mind advances that have been happening and the pace at which advance has been happening, that potentially there will be a method of doing the same analysis that isn't destructive in the future.
Yes.
It's a hard thing to figure out.
Yes. And you can also sometimes you can create archives so that you're not completely destroying. So for example, with our ancient DNA extractions from the dental calculus, we have the extractions preserved. So say we run a 16S rRNA analysis, or say we do next generation sequencing to look at what kinds of microorganisms are present down to genus level, or maybe even species level, there's still a lot of DNA preserved in the extractions. So they're sitting in the -80. And 10 years from now, 20 years from now, when techniques improve, even though the calculus from those specific teeth has been destroyed in order to do the extractions, there's still a lot of DNA left in the extractions themselves. And so as techniques improve, we'll be able to, save that material so that we're not destroying the entire sample, right?
Yeah, definitely. So you're looking at isotopes, you're looking at teeth, and you're looking both at occupational and dietary, but then you're also looking at these bacteria from this infectious spillover.
Yes. So our goal is, hopefully, we have extractions for 40 teeth. And this is a commingled assemblage, so it's not always possible to say that these are teeth from different individuals necessarily. But we have extractions for 40 teeth, and we got some exciting, preliminary data that there is ancient DNA preserved in the extractions. And in some cases, for at least 8 of them, it looks like it's a significant enough amount that we should be able to see something when we do the sequencing.
So you'll be able to identify the type of pathogen?
Yeah.
That's really exciting.
Yes, we just have to do the actual sequencing. So we haven't done that yet. We've been moving very, very slowly. The tech that I had working on it, Charlotte, she is an amazing person. She was an undergraduate here. She graduated in December, and I recruited her to come into my lab. She'd been trained to do microbiome research on cockroaches, to do DNA extraction and sequencing, and then a little bit of the bioinformatics as well. And so I sent her up to work with Laura Wyrick at Penn State, and she got trained in the clean room. And we used the clean room here. Laura came down and kind of inspected it and helped us figure out a protocol for our specific circumstances. And that was very generous of her. And then she also spent time with Charlotte in her lab, showing her the extraction protocols. And so it does look like she did the extractions in a very clean manner, and that, there is some ancient DNA preserved. So we're hoping when we go ahead and do the sequencing that we will be able to shed new light on the evolution of some of these pathogens.
That'll be super cool. And just a note for listeners, ancient DNA is exactly what it sounds like. It's just really, really, really old DNA. It's not like a different type of RNA or DNA. Like, the point is, it's just really old. And so you have to amplify it a few times when you're doing the extraction process so that you can actually read it. So there's all these techniques that have really improved in the last decade. It's very exciting stuff. Okay, so isotopes, teeth, ancient DNA analysis, bones. You also do bones.
Yeah. So we're using the micro CT on these individuals, as I said, to look at microstructural changes related to pathology. But we're also we have a lot of infants and children from archaeological sites. And my collaborators, and I have a whole bunch of collaborators, Megan Perry, Megan Brickley, Haley Wynn. We are looking to create an atlas of variation in, I'm sorry, I almost forgot my wonderful colleague from Drexel University, Haviva Goldman. Last but not least, anyway, we're seeking to create an atlas of variation for microstructure at the midshaft femur for infants and children. And basically, we would like to get a handle on what microstructural variation during ontogeny looks like, but then also have a reference point for evaluating infants and children who have osteopenia. Quite a while ago, for my dissertation research, I worked on these kids from Gown, infants and children, and they seemed to be stunted and wasted. They had short stature for age and low body mass for stature. And Haviva and I actually sectioned the femurs from these infants and children, and we looked at their microstructural characteristics quite a while ago. And in 2014, we published a paper suggesting that you can use the volume of pores and their distribution to look at some of the causes of biocultural stress and pathophysiological variation in past human remains and distinguish things like infection or malnutrition from activity level at the midshaft. So that's a whole long story. I'm not even going to go into it. It's a very, very long story. But at this point, Megan Brickley and Haley had published a paper questioning whether our results actually represented pathological conditions in these infants or whether it was actually just part of the range of variation. And so I contacted them and we got together and we've scanned in the micro CT 120 infants and children from sites in Jordan and France and some contemporary remains from Australia at this point. And basically, we decided what we really need to do is not argue about these 14 samples from 3,500 years ago. Let's actually try to, at this point, now that we have the technology and we have the ability, let's try to understand what variation actually does look like and how that can become deranged in a small proportion of infants and children. So
yeah.
Working on it now, and we're hoping to expand that sample and do more with that too.
Yeah, that's amazing. I think that there's sometimes there's this idea that everything has been done, but this is a great example of you are trying to figure out literally what is the natural range of variation in femurs in subadults.
Yes, because if you think about it, so I often thought that as an undergrad, I would read the literature and I'd be like, wow, we know so much about this and that. But the more you read, the more you realize, right? That's the source of questions is reading. The more you read, the more you realize how much we don't know. And in this case, if you think about what we can know about human skeletons from living infants and children, it's primarily based on remote sensing, right? There are large samples of X-rays that were collected before people realized that, that kind of radiation is not good for infants and children. So we have some X-rays that were unfortunately obtained quite a long time ago. We have other types of remote sensing that we can use today, like DEXA or ultrasound to evaluate bone quality. But really what we're looking at there is just bone mass in general, right?
Mm-hmm.
It's not the structure, the microstructure of bone. And you're not going to take biopsies from healthy, typically developing children. And those types of human remains don't typically end up in cadaver collections either. And even if they do end up in the few limited cadaver collections that exist from infants and children, you often can't do destructive analysis. So we actually, even clinically, don't have a very good sense of this range of variation or what the process of derangement really looks like in terms of microstructure. So it's something we're pretty excited about being able to address using archaeological bone.
Yeah. And the process of derangement, as you call it, a lot of that goes back to what you were talking about earlier, which is pathology. So developmental problems, environmental factors that might mean a lack of nutrition. There's so many things that could cause these sorts of things. I think probably any listener who has read anything from Charles Dickens is familiar with something like rickets, which is, for example, something that you might be able to see, or that you would be able to see in this kind of testing.
Right.
This is super cool. And one of the things that I've been kind of circling around and driving at as I've been talking to you is how holistic your research is. I feel like I've spoken to many biological anthropologists that are like, I am looking at teeth. I am looking at, and that is fine. There's a lot to know about teeth.
Yeah. Oh, yeah.
Obviously, you are looking at teeth as well. This is actually kind of my year of the dental anthropologist. You're the fifth dental anthropologist.
You can't ignore teeth. They're very important.
I know. I every time I go to the dentist, I'm like, you know what I learned recently? And they're like, I'm sure there must be a note in my chart that's like, do not ask her about archaeology.
Yeah. Yeah, I find dentists actually find it pretty fascinating, though. And I have a lot of students here at UNCG Biology who are pre-dental and super fascinated.
Yeah.
I have a group of students right now who are pre-dental who are leading the project on cementum annulations and discovering that, sectioning contemporary, well-preserved teeth, even from a forensic context, even teeth that are, less than 50 years of age since the time of death, sectioning is very easy and it's really straightforward to look at these cementum annulations. And when they were learning how to do the sectioning, I'm sure that they thought that this project was going to be really easy. But once you start working with archaeological remains, hard tissue histology can become very complicated very quickly because of diagenesis and other taphonomic factors. And so now they've gotten very, very deep in the rabbit hole of not just looking at age at death from the cementum annulations, but also trying to determine if there are ways that we can detect diagenesis prior to sectioning so that you don't end up in circumstances which, some people who've tried to work on cementum annulations have published papers like, yes, 80% of our sample had to be thrown out because the annulations were not visible. And, those kinds of taphonomic challenges should be detectable, hopefully, from the surface so that we don't have to end up doing a destructive analysis and finding out that we're not going to be able to get the kind of data that we were looking for. So, yeah, I have 3 undergrads that are working on that project, and they're all pre-dental and they're super excited. And one of them is actually considering doing a master's degree here before she goes to dental school because she's so excited about doing this project, which is awesome. They've been doing an amazing job.
It does. One of the things I really like about anthropology is that, like your colleague Sangeeta.
Mm-hmm.
Or Jeremy De Silva was telling me about a colleague that used to be a podiatrist. It's like people who've had other careers and they're like, you know what? I want to know about what was happening in the past. So that why did we end up here?
Well, and that's one of the most exciting things for me about teaching in a biology department is getting to contribute anthropological perspectives to people who are training to go to medical school or to be dentists, or, I teach courses on evolutionary medicine and human growth and development. And I also teach a course on the ethics of anatomical sciences, including biological anthropology and the history of dissection, and how the ethical challenges and the, historical injustices have shaped medicine. And it's so critical for these students to have access to this information. And, they get really, really deeply involved in learning about it, because this has a huge impact on levels of trust for the medical establishment, who ends up becoming part of the medical establishment or medical professionals, how patients are viewed, how patients can access healthcare or not.
Mm-hmm.
It's a really important aspect, and there should be more biological anthropologists who have the ability to actually teach anthropological content in pre-professional programs.
Yeah, this is definitely a theme that I see throughout your written work as well. One of the things that you focus on is the ability, like the contributions that biological anthropology has to modern problems, because a lot of people hear like, oh, I'm looking at a Indus River civilization from 3,500 years ago, and they're like, whatever. But hopefully not the people listening to this podcast, though.
Well, yeah, but I'm very interested in relevance. I'm interested in thinking about contemporary challenges, like ethical challenges, like climate change, from an anthropological perspective, because we actually have so much to offer. So, like Jane Bykster and I and Betsy Ewell are developing a workshop. We're developing a workshop on 1 paleopathology in advance of the evolutionary medicine meetings in Durham, and submitted for funding for that. And, one of the major things that I hope will come out of that is interdisciplinary grant proposals to really bring paleopathology and bioarchaeology into these larger realms of discourse, and for folks in our field to make their contribution, because the stuff that we do, people in professional programs, people in STEM in general, people in policy, people in finance, the people who actually run things, they're super interested in what we do. And it does have a lot of relevance for the questions that they are interested in. It's just a matter of communicating.
Yeah, for listeners who are interested, there's 2 articles that are fairly recent that you wrote, or that you are a co-author on, that I think really highlight this perspective. One is Climate Change, Human Health and Resilience in the Holocene. And the other is Building a Bioarchaeology of Pandemic, Epidemic, and Syndemic Diseases, Lessons for Understanding COVID-19, both of which are readily available. Again, I congratulate you on that. Every time it's easy to get articles, I thank the person that I'm speaking to.
I always try to make them available because I really hate paywalls. Science should be open access. It just should be.
I totally agree. And if you ask scientists, almost all of them say the same thing. And the ones that don't want to share their data, I'm not going to name names, but it's fishy as hell.
Yeah, sure.
But yeah, so it's one of the things that you like explicitly say is that anthropological insights, this is a quote, are not consistently incorporated into human security literature or policy and planning efforts, which can be based on potentially dangerous misconceptions about human evolution. And that is something that I see in my day-to-day career. Like, I had someone at work the other day say something like, I'm glad I got braces as a kid because now my kids will have straight teeth. And I'm like, that is straight up, that is Lamarckian. Like, what is going on? And that's like such a minor example. And sure, that person isn't making policy decisions about like what to do with climate change. But I think like
they are making decisions in the world.
They are
right?
making decisions in the world, and I work in finance. Like,
yeah. So actually, finance is what got me interested in relevance, because this is very random, but I was in Manhattan back in 2014, maybe. It was a long time ago. It seems like a blink of an eye to me, but I was in Manhattan to give a talk at CUNY. And I went to a party, and I'm not going to tell you how I ended up going to this party because it's a name names situation. So I'm not going to say, okay. But I went to this party. It was a Super Bowl party, and there were a lot of Wall Street folks at this party. And, you know, I didn't really fit in at this party, but I had a familial type of connection. I'll say that. And so I was there, and folks were, talking about what they're reading and what they are interested in. And someone made the mistake of bringing up Steven Pinker and talking about how great and brilliant Steven Pinker is. And we got into a very long conversation about Steven Pinker. And I had very recently, right before that, like a year before that, been quoted in a paper in Science saying that Steven Pinker cherry picks his data, which was ironic because I actually wrote this 10-page email to the reporter about Steven Pinker and the problems with his analyses. And that was literally the only quote that they put into
cherry picked.
They cherry picked my
quote
email.
About cherry picking.
Yeah. Anyway, so that's actually what got me interested in this in the first place, is that I realized at that party that, these folks have unbelievable access. They live in a world that is unimaginable. And this was brought home to me again when I received an award for my research, and I was invited to attend the Clinton Global Foundation meeting this past September. And, there are a lot of policymakers there. There were a lot of indigenous activists there, which was amazing. But there were also, these financiers. And again, talking with them at lunch or in just different spaces at this meeting and thinking about, the kinds of misconceptions that they have that come from reading. I mean, they're reading.
Right.
They're intelligent people. They want to understand the world. But unfortunately, the people who provide quote unquote anthropological knowledge to the popular audience are not anthropologists. Steven Pinker, Jared Diamond.
If I go on one more date with a guy who tells me that Sapiens is the pinnacle of anthropological achievement, I will cry.
Yeah, it's disturbing. And so, you live in this, as an academic, you live in a bubble. And when I went to that party, it was, I guess, 10 years ago almost now. It was in October, but it just sort of opened this whole window for me. It made me realize, I don't have the right to just be in my little bubble.
Mm-hmm.
It's actually not okay, because people suffer and die because of these policy decisions. And when we're trying to work for a more equitable and sustainable future, and especially given the challenges of climate change, which are really devastating, it's not okay for me to just be in my little bubble of I like teeth. It's just not for me. For me personally, it's just not acceptable. And so that's sort of where things started to shift for me. And I didn't really see how I could make a contribution immediately. It took me a few years. I had to go on a sabbatical and sit in my bathtub and think about how can I actually shift things. Once I became a full professor and had the privilege of sort of doing whatever I wanted, I got a sabbatical, and I literally spent that sabbatical reading and sitting in the bathtub and, publishing and stuff too, but also just literally thinking, like, what am I going to do in order to live the values that I have? Because I really do want to communicate to a broader audience. And I really do think that it's critical.
Yeah.
And that anthropology has to be part of these conversations. And so that's sort of where all of that kind of commitment came from.
I think one of the things that I've realized is that people are interested in anthropology. They just don't realize they are.
Yeah.
And what is publicly available to a lot of people, that kind of like surface level, typically it's like a historian or economist that's like repackaging stuff, is not really going to do it. But reaching those people can be difficult. That's something I think about a lot.
Yes. And so recently we've been like super fortunate to have some really great anthropology communicators, right? Like Jennifer Raff. Her work is absolutely astonishingly good, right? And she is communicating incredibly complex, difficult concepts about ancient DNA and what it's like to do lab work in that field. And she is a brilliant communicator. Her writing is incredible. And she can write a popular book that anybody could read and enjoy and appreciate. And of course, she's getting recognition for that, which she very richly deserves. Not all of us can write for a popular, right?
Yeah.
I wish that I could do that. Maybe I could try harder. I don't know. Jennifer Raff and others do it. And so luckily that's not a niche that I feel obligated to fill anymore. Not that I ever did fill it. I never did. But, in the past I had wished, I wish I could, write a book. And I did start a popular book. But I'm not that good at communicating in that kind of writing. Like that's a very special skill. And luckily, we do have people in our field who do that, and they do it really well. So that's awesome. But for me, it was more like I can communicate in a scholarly way. I can write a scholarly article. And so I can make the audience broader. I can bring anthropology into scholarly discourse beyond the bounds of anthropology. And we can communicate with policymakers. We can communicate with people across STEM. And I feel like there are a lot of people who are also doing that really well at this point, too. And so I'm sort of part of a large community of people who are achieving those goals. And that's really exciting.
I think that's wonderful. And there is a different differentiation. And this is something I do say all the time is you have to know your audience, right? Like pop anthropology, that's great. The more people that know about it, that's wonderful. It might trigger something in a policymaker, but like ultimately when you're talking to a policymaker about making decisions, they're going to want something more academic, which is something that you provide. I really recommend listeners go and look at those articles. I'll make sure to link them in the episode notes so that they can take a look. But it is 156/456.
Yeah.
And I want to make sure that you can go do other things with your life. So are you ready? Is there anything else that you wanted to say that you didn't get to say?
Oh, gosh.
I know there's always so much.
Yeah. Well, we covered a lot of ground.
Mm-hmm.
And so I feel like you did a really good job of keeping me from just rambling about random topics.
I will always take a compliment. Thank you.
It's not an easy task, but I think you did.
It's a good thing you're so interesting. Okay, so are you ready for the last 3 questions then?
Sure.
Okay. So the first question is, if someone wants to learn more about the kind of work you do, where is a good place for them to start?
So I looked at those last 3 questions, which you sent me in advance, and it was so hard for me to pick like a single place that folks could go. But here's what I will say.
Okay.
There are some really, really good overview books for like undergraduate students or entry-level grad students who are getting started in the field. For example, the companion to paleopathology, Anne Grauer's recent book, really, really great book. And it covers so many topics. I mean, that's the thing is when I think about a question, an introductory text, well, introductory to what, right? Because there are so many topics in our field. It is such a holistic discipline. So that particular book I really admire. And I also really admire Jane Bykstra's recent revision, Ortner 3, basically, of an introduction to human skeletal pathology. That Ortner 3 book is incredible. It's an edited volume. Again, both of them are edited volumes. And she really pulled together experts from all across paleopathology. And you can really get a good introduction to any kind of pathological condition and also to issues surrounding pathology. You know, if you really want to dive into pathology, you know, you got to get into like those multi-volume, like this down here. I've got my Resnick's diagnosis of bone and joint disorders. I have a copy here, a copy in the lab, and I copy at home just so that if I ever want to read anything about bone and joint disorders, I can. You know, that's a little bit different level. But if you want an introduction, I really recommend those two books. They're so good.
That is perfect. That is an excellent answer. The second question I have for you is, what are you reading for work right now?
So right now I am reading about planetary health, which is a discipline that is similar in concept to One Health or One Medicine, but it's actually specifically focused on making suggestions for policy. And it's specifically focused, unlike One Health, where, you know, you're thinking about the ecosystem and plants and animals as equivalently important to humans. I'm really sorry to say this, but planetary health privileges human health within that scheme because, you know, obviously all important, like ecosystem health, environmental health, other plants and animals, you know, we have to have everything, right? Everything has to be healthy in our sustainable future, right?
Right.
However, planetary health has a specific focus on what can we take from a One Health framework that specifically informs policymakers about humans and our future and sustainable societies and sustainable development. So I've been reading a lot about planetary health, and I'm actually writing a paper for a book through Springer Press that's on planetary health about malaria. So I've been reading a lot about that framework and really coming to appreciate their perspective.
Excellent. Another good answer. One Health is a concept that was recently introduced to me via your articles. And then I'm also reading a book called The Human Disease by Sabrina Schultz out of the National Museum of Natural History in DC,
Yes.
Smithsonian Museum.
Awesome.
It's so interesting. That is like not kind of the avenue that I took when I was in anthropology. So it's been like a whole new world for me.
Yes. And that is an amazing book. And she's an amazing scholar too.
Yeah. The book is, I have learned so much. I have become insufferable at dinner parties. So especially last night, my best friend's father took us out to dinner, and I spent at least 10 minutes talking to him about Ebola and the transmission of Ebola and like not taking into account cultural practices.
Excellent.
And he was like, I really want to eat my fried chicken.
You are upsetting me.
Like maybe after dinner. My third question is, what are you consuming for fun right now?
So I really enjoy kayaking. And I love kayaking with a group of friends here in Greensboro. They are an amazing group of women. Most of them are actually kayaking instructors. And so it's super fun to go kayaking with them. They also do kayak camping, and I go with them sometimes. And I really love it. It's so relaxing. And I learn so much from them about everything, but also about kayaking and camping. And so that's really, really fun, and it gets me outside and I get exercise and get back into nature. And it's so important to get out of your office and away from your computer and be outside with friends.
Yeah. That like groundedness that you get is super important. My therapist told me that, so that's a little bit of free therapy for all the listeners. Well, on that note, thank you so much for coming to talk to us, and I hope you have a great day.
Thank you so much.
Thanks for joining me for this episode of the Anthrobiology podcast. If you have questions or comments, you can reach me at gabby.lapera@anthrobiology.com. That's spelled GABY.LAPERA@anthrobiology.com. This show is produced by me. Music courtesy of Chad Crouch, aka Poddington Bear at Sound of Picture.